So it is a pleasure for us to have today in our Bar-Ilan University Vision Science Seminar, Professor Paola Binda from the University of Pisa. Professor Binda did her bachelor's degree in psychology at USR Milano, graduating with honors, and then continued to a master's degree in cognitive neuroscience, also graduating with honors. She then did a PhD there in neuroscience biotechnology under the supervision of Professor Maria Concetta Morone. She was a Marie Curie postdoc fellow at University of Washington, Seattle, and then a senior postdoc at the University of Pisa, Italy, where she is now faculty at the Department of Translational Research and New Surgical and Medical Technologies. She published dozens of papers, many of them in high-profile, peer-reviewed journals as Current Biology, Journal of Neuroscience, Nature Communications, Trends in Cognitive Sciences, Annual Reviews of Vision Science, and more. She also published 2 book chapters, and she was elected to be a member of the VSS Board of Directors recently. She has organized several scientific meetings, and her work is funded by the prestigious ERC Starting Grant as well as by additional grants. She has given many invited talks across multiple countries. Her research focuses on the physiology of the human visual system, which she views as a model to understand brain function in general. In her research, she uses psychophysical methods, gaze and pupil tracking, and neuroimaging techniques, including ultra-high field fMRI. In her work, she also examines inter-individual variability and its modulation by changes. So without further ado, I'd like to welcome you, Paola, to our seminar to hear about your exciting research, and thank you so much for joining us today.
Thanks to you. Thank you for having me. I'll take a second to share my screen and start my presentation, which is here. Okay. So the story I'd like to tell you today is a collection of a few different approaches that we've been developing recently in the lab, some of which I've recently reread based on a very different literature. We're working on the visual system sensory neuroscience, but I've come to realize that a lot of the things that we are looking at have to do with heuristics, biases, and maybe reread in the light of the work of Nobel laureate Daniel Kahneman and his colleague Amos Tversky. And specifically, the kind of heuristics, I mean, I guess all of you know all about this, but maybe some of the people don't know, and I'd like to make an example. So this is an example of a decision-making situation where our behavior, our decisions are absolutely irrational. So we hear about this Linda, who is now 31 years old. She's a single, outspoken, and very bright. She majored in philosophy. As a student, she was deeply concerned with issues of discrimination, social justice, and also participated in anti-nuclear demonstrations. So which is more probable that today she is a bank teller, or is she a bank teller and an active member of the feminist movement? Obviously, number 1 is the right answer, but also obviously there is something in us that tells us that's just impossible, that the second option is a lot more reasonable. And this has to do with the schemes that we apply to our understanding of reality. And those schemes are sometimes too rigid to fit the actual reality, which is how people change from when they were students to when they become an adult bank teller. And that's something that may also apply to the sensory environments, which I would suggest are a lot more stable than people's ambitions and life goals. What I'm saying here is that heuristics and biases shape our decisions, but they also shape our perception. I'm not saying anything odd here. Here is a very famous example of the hollow mask illusion taken by from the work of Richard Gregory. So we are looking at this 3D shape, and it is very hard for us to see it as a convex shape. we always see it as a concave shape. I'm getting this wrong, sorry. As bumping outside. And that is because a lifetime of experience with faces tells us that faces are not inward, but they are always facing outward, okay? So this is one example of, we can call it heuristics, we can call it a priori information, that we use and shape our perception in a very profound way. Now, these heuristics reflect contingencies that we encounter in the sensory streams. And we can study the acquisition of these heuristics. That's the first bit that I'd like to talk to you about. Using many different methodologies, including pupillometry, perhaps another point of contact with the work of Kahneman and Stretsky, who also introduced this technology to study decision-making. We'll use that to study the implicit learning of these contingencies and how we come to know that faces are a certain shape. We'll then move on to other examples of very basic sensory neuroscience, where I think we can reinterpret, reframe something that has been considered and classified in the category of plasticity, specifically short-term plasticity, in the category of failed heuristics. And we can use these to understand something more about the brain circuits that support heuristic-based perception and plasticity, and possibly also to open new ways to trigger, to support brain reorganization, again, based on these reset of heuristics. So this is the three categories of studies that I'd like to tell you a little bit about. And starting with the contingencies. So here is one, I've already said, the hollow mask illusion, but there is also many other examples. So think of yourself being at home at a friend's and going through the pages of their family book, family album. There's going to be lots of different faces. And even as you page through this book completely thinking about something else and passively exposing yourself to this stream of visual information, there is a lot that you're learning from these. And specifically, there might be a co-occurrence of two faces, and even if you don't know anything about this family or those faces, you'll come to realize that those are related. Those are a relation, maybe the mother and the son, the mother and the daughter. This type of contingencies are continuously acquired, and they can be acquired implicitly. There is a whole literature out there about this form of statistical learning, also known as implicit learning. We learn a lot about this, but there's also many things we don't know about it, which are mainly related with the methodological limitations, because once you ask about what you've learned from these pictures, you've kind of made the person non-naive anymore, and you've interfered with the learning. So we thought that using pupillometry, we could contribute to this field because we can monitor what happens to the processing of the stimuli without ever interfering with your conscious elaboration, conscious processing of the material. So we did not work with the pictures because you know that the pupil responds to light and it's sensitive to lots of other basic visual features. So we had to strip down a lot of these interesting characteristics and used much more boring stimuli. The stimuli look like this. This is a set of bars that show up on the screen. They may be of different numerosity, different orientation, but the luminosity of the display is always exactly the same. And there's two sequences of stimuli. This was one, and here comes the second one. And they probably look very similar to you. And this is one of the interesting facts. They look similar, but they are very different in terms of their temporal structure, temporal statistical structure. So before I look into that, let me show you the basics of the stimulus and the pupil responses that they elicit. So we showed the stimuli at a pace of 2 per second. So every 500 milliseconds was a new stimulus. And I'm marking them with just a number of items on the screen. Because the stimuli were coming up at a regular frequency, 2 hertz, we saw oscillations in the pupil diameter, which are very tiny. Look at the units of measurements if you wish. But nonetheless, very systematic. And you can clearly see that in the Fourier spectrum. Now, this was the first sequence that I showed you, where the temporal sequence of events was completely random. However, we also had a second sequence, the second that I showed you, where we imposed a statistical dependency of in the temporal structure of the stimuli, such that every time a screen with 2 items showed up, the following stimulus would have exactly 12 items. Every time a 4 showed up, the following one would have 50, and 24 was always followed by 6. There were also rarer pairs that I won't tell you more about. So this means that there were pairs of stimuli. The sequence of the pairs was still randomized, but within each pair, the occurrence of one stimulus predicted with 100% accuracy the occurrence of the second stimulus in that pair. And what we saw is that the pattern, the oscillations of the pupil diameter was completely different than before, even though the stimuli were exactly the same. They were just presented in a different order. And these oscillations clearly show the second peak when analyzed in the Fourier spectrum, in the frequency spectrum, which was the frequency of the pairs. So there were 2 stimuli per second, 1 pair per second in this second structured sequence, and the pupil was responding to this pairing frequency. We did a little bit more to quantify this effect. Number one, we looked at how long did it take for you to establish this pupil oscillation, this pupil response to the statistical structure. So we quantified the amount of energy in the pairing frequency normalized to the amount of energy at the same frequency in the random situation. That was a way of reducing interindividual variability. And then we looked at this as a function of time. So here I'm looking at the energy at the pairing frequency as a function of time from the beginning of the experiment. And we see that this log ratio is very close to 0, meaning that there is no pupil response to the pairing at the beginning, but this gradually accumulates over time, and it becomes significant round about 2 to 3 minutes after the beginning of the experiment. And because we only had few pairs, few structures in our stimulus, this means that it only took about 40 to 50 repetitions of the pairs in order for us to, in order for the pupil to show that we had learned, that we had implicitly internalized the sequence, the fact that 12 always followed 2, etc., etc. The other thing that we did was to play around with different types of stimuli and different types of task demands. In one situation, participants were paying attention to the numerosity of items on the screen because they were supposed to hit the button every time the same numerosity was presented twice in a row. And this happened, I'll go back one slide for a second, when these rare pairs were presented. So for example, a 12 was followed by another 12 and a 99. And we introduced these pairs to have the repetitions without breaking the regular periodicity of the pairs. Okay, so in one condition, they were paying attention to the numerosity. In another condition, they were just paying attention to the orientation. They would hit the button every time. The same orientation would be presented twice, irrespectively of how many bars there were on the screen. The results of these two conditions are presented in the first two bars, and they are essentially the same. They are indistinguishable, meaning that passively viewing the stimuli did not prevent us from learning, or at least did not prevent the pupil from showing this signature of implicit learning. And it also didn't matter which materials we worked with. So we could work with digits instead of arrays of bars. We could mix the digit and arrays, so symbolic and non-symbolic stimuli, and we would still get the same magnitude of the effect. So what is happening here? First of all, the pupil knows something that we don't, which is weird, but it shows that the pupil dilation detects the temporal structure in a stimulus, even though we as participants could not tell the first type of sequence with random temporal order from the second one with the structured temporal order. And that's not because the pupil is smarter than us, but it's possibly because the diameter of the pupil reflects the dynamics of neuromodulators, neurotransmitters that are involved in the acquisition of implicitly learned information. So the way I see this, and this is just a very super simplified toy model, is that there is some response that is elicited in the brain or even at the level of the pupil diameter by the stimuli, each stimulus in the pair. And then there is a bout of a novelty response, a bout of brain response every time something new happens, which is the occurrence, corresponds with the occurrence of the first stimulus in each pair. And this novelty response could potentially be linked with activity in the very well-known locus ceruleus nucleus, which sits in the brainstem and releases norepinephrine across the brain. Norepinephrine has lots and lots of effects, including enhancing sensitivity to sensory stimuli. And one effect is also dilating of the pupil. So we are looking at the pupil as an indicator of changed processing of sensory information. So this brings us very close to a slightly different concept, a slightly different way of seeing this dynamic, which is usually termed predictive coding, but it is very similar. The idea that every time something surprising happens, we have a strong response. Responses to unpredictable stimuli are strong. And every time something very predictable happens, we have less of a response. If the second stimulus comes up, I already know which stimulus will be, and its response will be suppressed. I'm saying nothing new here. Now, we have noticed a resemblance between this pattern and a very different phenomenon. This might sound a little new to you, which has been interpreted in a very different way. And this phenomenon is the phenomenon of homeostatic plasticity or short-term plasticity. And I'm using this graphical representation in the hope of calling your attention to the similarity between these two concepts. So in homeostatic plasticity, we are seeing a boost of the response to a deprived sensory channel, for example, a deprived eye. And I'm going to suggest that this boost could also be linked with that channel becoming transiently unpredictable. So this is the key of the story. But let me tell you a little bit about this phenomenon first and introduce it. So this phenomenon has been thoroughly investigated since 2011, when a colleague of mine, Claudia Lunghi, who is pictured up here as well as here, started studying the effects of applying a patch over one eye of normally sighted adult visual participants, basically us in the lab, for just a couple of hours. And this simple manipulation, somewhat surprisingly, changed one of the most basic features of the visual system, which is ocular dominance. So here is an example result and one of the methodologies that was used to quantify the effect. The methodology is binocular rivalry, which we can use to get a very precise quantitative measure of which eye is dominating over the other and by how much it is dominating. So we present two different stimuli to the two eyes. Perception alternates between them as represented down here. And the amount of time that one eye dominates is a good measure of its relative dominance, of its perceptual dominance. So we can take a measure of how much one eye dominates over the other before the manipulation, and then apply this simple patch for 2 hours, remove the patch, measure binocular rivalry again, and what we see is that there is a shift of ocular dominance, very prominent and significant, that goes in the direction of the patched eye. Now, this was very counterintuitive at the beginning because we came for a whole history of physiology where closing one eye, depriving one eye during development for a fairly long period of time would lead to a suppression of the deprived eye, which is what we call amblyopia. Here we're actually seeing the opposite in terms of sign. We are seeing a boost of the deprived eye. And we're also seeing a behavior which is transient. This boost is seen for a short period of time, around about the same duration of the manipulation, when we talk about significance at least, that vanishes over the course of the day, basically. Amblyopia, on the other hand, is a persistent deficit, which we may wish to intervene upon, and I'll come back to this at the end of my talk. So anyway, we have this very obvious shift of ocular dominance that boosts the dominance of the patched eye. This can be seen in binocular rivalry, but it has been seen in with many other techniques, and we contributed to that using ultra-high field fMRI. We measured visually evoked responses to stimuli presented in either one eye or the other before or after the application of of that patch for 2 hours. So here is a map of visually evoked activity, and it's just meant to orient you about where we are in the brain. The activity is focused in the occipital lobe and the thalamus, and we can visualize that on the flat map of the cortical surface. And then we can take a measure of the difference between this pattern of activity elicited when one eye or the other is stimulated. And if we do that before the manipulation, nothing really shows, meaning that the two eyes were balanced, entirely expected, given that we are working with normally sighted, non-amblyopic participants. But if we did that again after the 2-hour manipulation, we saw a very obvious shift of activity, very strong, much stronger activity in response to the deprived eye compared to the non-deprived eye. Okay, so there's multiple ways of interpreting these, and one of these is the concept of homeostasis. So one eye has not been fed with a signal for a short period of time, and the brain responds to that by increasing the gain, cranking up the volume to try and listen to it better. We thought that there could be a very different interpretation of the same phenomenon in terms of expectations. So we've grown our entire life expecting that the two eyes give comparable information. And that's the very basis for things like stereopsis. Now, suddenly, one eye fails to signal, to give signals compatible with the other eye. And it fails to signal events that I know are happening, for example, because I'm making them happen, because I'm playing with my hands. So this failure of expectations could also contribute to this boost of responses in the framework of predictive coding. Now, to test this, we wanted to have a situation where we are not playing with the strength of the signal, so we are not depriving the signal in either eye, but we are playing with the expectations linked with signals in other eye. And for this, we moved to a much more sophisticated manipulation. So we were not using a simple touch anymore, but we had to move into an altered reality environment where people were wearing virtual reality goggles, the Oculus set, but they were using those to see their own visual field. And that's because a 3D camera a stereo camera was mounted on top of the visor. So the situation was basically this. The participants was seeing his reality or their hair reality through the goggles. And this allowed us to change one of the two images, one monocular image, in a systematic way. We simply delayed it by a long time, by 1/3 of a second. And what the participants saw was something like this. So the two eyes were carrying exactly the same amount of information. Neither of them was deprived, but one of them was always too late to help with behavior. So you'll notice that participants were engaged in this visual motor coordination task. It was actually very simple. They were building towers with blocks, and they were meant to make towers as tall as possible without making them fall. And one eye, the online eye, was carrying useful information. The other eye was actually harmful because it was signaling the end in a place where it wasn't at that time. So before and after this manipulation, we measured binocular rivalry, exactly the same as was done before with monocular patching. And the manipulation here was only 60 minutes. So we reduced the time from 2 hours to 1 hour. And what we saw was that, again, ocular dominance shifted, and it shifted in favor of the manipulated eye, the delayed eye. So failing expectations was having a similar effect as reducing the signal altogether, as depriving the signal altogether. We had several controls. One of these was a situation where participants were passively watching a video, somebody else playing the same task, but this time they were not actively engaging in any of those changes. And in this situation, there was no effect. You can say, but this is not exactly the same as in the active situation. And to try and answer this, we had another replication of this experiment, where again, we had in a separate set of participants, the active monocular delay situation, same as I showed you before. But during this, we recorded everything that happened during the experiment. So the video of both eyes, the online eye and the delayed eye. And then we replayed this video to the same participant for the same amount of time, still 60 minutes, in a passive situation. So they saw exactly the same visual stimulation. Nonetheless, there was no ocular dominance shift in this passive situation. And another interesting bit that we added here was that we replayed again for a second time, the same video. And this time we replaced the delayed image with a homogeneous gray screen, thereby reproducing the monocular deprivation situation, although we didn't achieve it with a patch, but with a screen gray out. And this allowed us to have exactly the same situation in the other non-deprived eye. In this case, we obtained a healthy ocular dominance shift as expected. But what's perhaps interesting is that the magnitude of the effect for the gray out screen, so replicating the monocular deprivation, is essentially the same as the monocular delay situation in the active condition. And not only is the magnitude the same, but also there is a significant correlation across participants, indicating that the effect of monocular delay and the effect of monocular deprivation are very similar to one another. Are they the same? Are they supported by the same mechanisms? Obviously, we cannot tell, but we have strong indications that they behave in a very coherent way, that we can at least mimic the effect of taking away the signal from one eye by simply making that signal unexpected or not useful for your behavior. Now, to interpret this, and here we are stepping into the neuroimaging part, the obvious reference is this kind of picture that shows up in the Candel Fundamentals of Neuroscience. It also shows in a beautiful review that I'm referencing here, the idea that even the simplest visual features, we're thinking of your dominance here, that are presumably set at the level of the primary visual cortex V1, are actually the result of recurrent computations, recurrent exchanges of information among multiple visual areas, whereby there is a hierarchy of feed-forward information that goes from V1 to multiple areas, but there is also a feedback source of information that goes back even to V1 and shapes processing based on contextual information. What's interesting here is that there is a specific brain region called the pulvinar in the thalamus that plays the role of a hub. It collects information from multiple areas, including the visual areas, and it sends back information to the very same visual areas, including primary visual cortex V1. So we thought that if the short-term plasticity effect, the effect produced by monocular patching as well as by monocular delay or other more complex manipulations, has to do with a case of failed heuristics, of a mismatch between the incoming information from each eye and the expectations. Incoming information being the delay image or the missing image from the patched eye, then this communication may be interfered with. To test this, we performed another fMRI experiment still at the ultra-high field magnetic resonance center here in Pisa. But this time we were not looking at the visually evoked responses. We already know that those are modulated and we know how they are modulated. This time we are looking at the resting state connectivity, meaning we are looking at the way areas communicate with each other even when they are not stimulated at all because the participants have their eyes closed. So we specifically focused on the pulvinar, as I mentioned, here highlighted in blue within the thalamus. And we also, as an obvious counterpart, as an obvious control, looked at the lateral geniculate nucleus, which sits right next to the pulvinar, but obviously plays a very different functional role because the lateral geniculate nucleus primarily sends feed-forward information from the thalamus to the primary visual cortex. Whereas, let me remind you, the pulvinar acts as a hub, collecting information from the cortex and sending information back to the cortex. So even though they are one next to the other, these two are very different physiologically speaking. And the question is, will they behave differently when comparing their connectivity before and after the 2 hours, 2-hour patching? So the answer is summarized in this slide, the first part at least, where we are looking at the resting state connectivity of the pulvinar. This is the resting state connectivity before the deprivation, which shows exactly the pattern you would have expected based on that figure that I showed you before. So the pulvinar is strongly connected with the entire occipital pole, as well as other regions on the lateral side of the brain. Now look at what happens after the monocular deprivation. With just those 2 hours of patch application, there is a strong reduction of connectivity of the pulvinar with the occipital cortex, including the primary visual cortex V1. And this is just a quantification showing the effect pulvinar V1 functional connectivity for the individual participants. We can have the same picture. I actually don't have it here for the LGN, and it shows nothing. The LGN connectivity is primarily with V1, and it is the same before and after the deprivation. We pushed a little bit forward and looked at a higher resolution, higher temporal resolution fMRI sequence, which allowed us to estimate the effective connectivity, an estimate of the directionality of communication, not just whether the pulvinar is communicating with the cortex, but which direction is this communication going. And to summarize the results of this dynamic causal modeling approach, we have that after the 2-hour patching, communication from the pulvinar to V1 was significantly decreased. There was no such change, neither for the communication from LGN to V1, nor for communication in the opposite direction. If anything, V1 communicated a little bit more to these subcortical regions. So the main effect that we are seeing is that the pulvinar stops sending information to V1. And what information is carried through the pulvinar? It is information conveyed from the other higher level cortical areas. It is contextual information. So what we're seeing here is that with 2 hours of patching, we're we're reducing the communication of contextual information to the primary visual cortex. Just to have a scheme of what we're saying, what I've been saying so far, and then move on to the last 5 minutes. Here is the way I'm sort of picturing this scheme. We have multiple areas that send expectations, P's for predictions in the light of predictive coding. And these predictions, these contextual signals are collected by a big hub, which is the pulvinar, which then sends it back to the cortex in general, and in particular V1, which we happen to be more interested in. If we were interested in higher level visual processing, we probably would have focused on another area, but the scheme would have been probably the same. This circuit, this scheme finds some support in literature from very diverse sources. There is literature on animal models suggesting that indeed the predictive signals are carried from the pulvinar to the V1, suggesting that this is carried as a form of inhibitory signal, which also fits with a lot of what we know about short-term plasticity and about predictive coding. But all these, and the last bit is, can we interrupt this circuit by introducing a mismatch between the predictions and the sensory signals, which would be one way of seeing that boost, of explaining that short-term plasticity effect in a very different vein as opposed to the classic, the standard, the homeostatic plasticity account. But more than having a different account of the same phenomenon, perhaps this scheme can inspire a different way of thinking about the stability of sensory processing. And if we can find a different way of interpreting this, maybe we can find also different ways of stimulating a reopening of plasticity. So here I'm going towards amblyopia and recovery of visual functions. The general framework I'm thinking about is, the general question I'm thinking about is, why is it that plasticity, at least in the sensory systems, is lost with age? So we've pretty much done with the first hypothesis that has been proved wrong in many ways, the idea that the brain loses the ability to change. That's clearly not the case. Although the brain keeps the ability to change, there is something that actively prevents it from changing, a sort of active stabilization. And I'm visualizing this as a fishing net, which is activated by mechanisms that we currently do not know. And the hypothesis, to bring a bit of the scheme before into this picture, it might be that what's drawing the fishing net is the pulvinar, which provides that stabilizing signal. Now, if the active stabilization hypothesis is true, then we might have ways of reopening plasticity even in the adult brain and even in the adult sensory cortex by interfering with the active stabilization. And now the question is, how can we do this? So even before, the question is, why do we want to do this? Why do we want to reopen plasticity? And here we go back to the standard model of monocular deprivation and amblyopia that I hinted at before. So the idea is that a prolonged period of monocular deprivation during development, depicted here, leads to a suppression of representation of core representations of the deprived eye. The same manipulation in adulthood does not produce the same effect, and we can visualize the curve of plasticity as peaking during a certain critical period. Although in adulthood we do not have this form of long-term plasticity, we've already seen that we do have a form of short-term monocular deprivation or short-term plasticity that I've talked about. So can we use this as a way of reopening plasticity and rehabilitating vision in this suppressed eye, in this amblyopic eye? So we're working with amblyopia, where abnormal vision in early development, just like in the kittens I showed you before, leads to a persistent acuity impairment, such that even if the disease itself is treated, the acuity of vision remains inferior in the formerly pathological eye. We asked whether repeated patching, intermittent patching in adults could improve the acuity of the amblyopic eye. And this time we're not patching the good eye, as people would have normally advised for, as it's standard in the treatment of children in the developmental age, but rather we are patching the amblyopic eye in an attempt to produce a mismatch between what is the system expecting, that this eye is not sending very good information, and what actually happens, that this eye is not sending any information at all. So the paradigm that we are using is taken from a previous study where we apply these repeated patching sessions combined with physical activity for reasons that I do not have time to go into. And we are measuring acuity in the amblyopic eye before and after this repeated training, this repeated patching. And we're also measuring fMRI responses. Very quickly, what we see is that there is an improvement of acuity. So by patching the amblyopic eye repeatedly in adult participants aged 25 on average, we do see an improvement of acuity. The improvement is not big, but it is significant. We can quantify these in terms of the standard Tamboli test. We can also quantify these in terms of binocular rivalry behavior. We are looking at amblyopic participants, so obviously before the training, we expect that in a binocular rivalry setup, they just see through their fellow eye and they see nothing from the other eye. After the training, this is no longer true, and we clearly see that, well, the amblyopic eye, to be fair, cannot dominate rivalry, but the dominance of the fellow eye has dramatically reduced, and they start seeing a mixture between the two eyes. And not only that, we are also looking, this is preliminary data, into their fMRI responses, which are clearly much reduced. These are the evoked responses in the visual cortex, much reduced for the amblyopic eye compared to the fellow eye. That's before the training, but after the training, we are seeing a very systematic shift, upward shift of the evoked responses in the primary visual cortex. And now obviously the question is, what happens in the LGN, what happens in the pulvinar, what happens to the communication between these areas? But I have to wrap up, and this is my final slide. So what we have started thinking is whether the plasticity of the adult visual system can be somehow revisited or reread through the lens of predictive coding. We know that heuristics, that's what I called it today, but implicitly learned contingencies in the stimulus stream are very important for shaping our perception. We are able to learn these throughout life, even just in a couple of minutes of presentations, as I showed you at the beginning. However, this is rarely needed because our sensory environment is really very stable, at least when it comes to basic visual properties like ocular dominance. Sensory plasticity, short-term sensory plasticity in adulthood, behaves maybe reframed as a rare case of failed heuristics, as a rare case where what we expect of the very basic visual features like ocular dominance is mismatched in the visual stream. And if this is true, then we can have a hint to understand the brain mechanisms that support this heuristic-based perception and also short-term plasticity. And I'd like to suggest that the pulvinar really has a key role in this. And we could also have new ways of reopening plasticity. I've shown you an experiment with patching the amblyopic eye, which is perhaps counterintuitive, but even more effective and perhaps even more counterintuitive could be to apply that monocular delay manipulation that we've used in the normally sighted adults to as a treatment for amblyopia. And I'm happy to discuss any of these. I'd just like to finish by thanking my funding bodies, my co-authors, and all of you for your attention. Thank you.
Thank you so much, Paola. This is really wonderful research, and I want to invite everybody to please unmute yourself and let's give Paola a big applause. Really great. And if you're happy to take questions, then I'll open the floor for. I mean, actually, I've got a question of my own, but I assume I won't be the only one. So I want to ask, with respect to the differences you found in the connectivity between LGN and V1 and the pulvinar and V1, I don't know how the anatomy of the pulvinar is, but I know that while V1 is like a map, so you could basically, you know, if you look into a voxel, it has a specific area in space. The LGN is much more, let's say, condensed. I always think of it as an egg. I don't know why, but it doesn't have, you know, a surfaced, precise map. And so I'm thinking about partial voluming or I mean, since it has the layers are mixed between the two eyes, you know, you've got, you know, the 6 layers and they come from each, both eyes, both the magno and the parvo. Maybe you're actually looking into the average of the 2 eyes. No, just the devil's advocate. I'm wondering if it may be the fact that you don't have, or maybe you do have, I don't know what the resolution is, what resolution is required in order to actually look at each specific layer, because I'm wondering maybe you will also see some aspects there. And also, I don't know if the pulvinar is or is not getting information combined from the 2 eyes, or it's keeping them separately. So these are things that I'm thinking about, and I don't have the answers to, but I'm wondering about these.
Yeah, no, thank you for bringing this point up. It is very important that we are never working at a resolution that would allow us to follow the alternation of ocular dominance bands in the cortex or the alternation of the ocular dominance layers in the LGN. What we're doing here is always having larger voxels, units of imaging, which means that we are mixing signals from the 2 eyes. This is, well, obviously a technical limitation on the one hand, but it's also telling us what we are looking at here. We are looking at a pulse signal, and whenever we see something that decreases or increases, it is distributed across those population of neurons. We still have, when we are looking at the visually evoked responses, we still have the ability to see that responses to one eye go up and responses to the other eye go down. But that is because at that particular moment, we're only stimulating one population of neurons or the other by using monocular stimulation. It would be fantastic to go further into the single eye representations, but these are short-term phenomena, and we are limited by the duration of the strong effect. So this is, like I said, a technical limitation, and we can't go there.
Okay, thank you. Very, very nice. Very interesting. Jeremy?
So, in your amblyopia treatment, do you get any recovery of stereo function?
Yes, we do. It's not dramatic, but in some of the participants that did not have stereopsis at all, they started to show a basic stereopsis at the TNO test. That's how we quantified it. It's more variable than the acuity change in the sense that it is more of an all or none phenomenon. Many patients did not have stereopsis to begin with, and they still don't have stereopsis at the end of the training.
Hagit, do you want to, is it okay to continue, Paula?
Of course, please.
Yeah, okay. So Hagit, do you want to unmute yourself?
Yes, thank you. So Paula, thank you for a great talk. I really enjoyed it. I'm considering the deprivation, at least through your talk, it seems like it's considered a binary feature. It's completely deprived or completely affected. I was wondering if you've considered something that would be more gradation, especially in the last part for amblyopia. Would there be an advantage of somehow putting in a gradient in the amount of deprivation?
Okay, so this is a very interesting point. We are currently compiling a meta-analysis of everything that has been done on this manipulation, because there's like 75 papers out there that looked at variations of the technique. The results are very consistent. The effect of gradually changing contrast is the one aspect that is not as clear. The results do suggest that this is a bit of a dichotomous all or none effect. So when you get to see through one eye, it doesn't really matter how much signal there is in there, which fits with the idea that I'm trying to suggest, that it is more of a matter of information rather than a matter of signal. But like I said, this is my own reading of a very small bit of data. In terms of amblyopia, I think I am completely, I completely agree with you that depriving is a very suboptimal way of going about this, because at the same time, we are trying to boost the signal in one eye by making it unpredictable, but we're also depriving it of the means of reacting to this. So my impression is that working with a technique like monocular delay, or maybe there are others that are simpler and more effective. I'm thinking prisms, for example, inverting prisms that also have been used. Could be a better way, because that way we would achieve both the unpredictability, the failed expectations for the amblyopic eye, and we would give it a signal for it to respond to.
So a delay would be possible to scale or to change its effect, right? I don't know how it would work with prisms. Prisms is also a binary kind of
Yeah.
effect. But yeah, thank you. Yeah, thank you.
Okay, lovely. Sarit also has a question, if that's okay.
Please.
Yeah, okay, Sarit.
So great talk, thank you. I wanted to ask also to extend Hagit's questions about different types of modulations. Have you thought of others beyond delay, beyond something that's consistent across the full visual field? That's my first question. And then another question is about the short-term plasticity versus the long-term. So these effects are really short-lived by the minutes. And I think Stefan Engel also has some studies like showing how also they diminish if you do this multiple times. So if you have a monocular deprivation or different types of adaptation, the effect of the adaptation is reduced after several sessions. So how do you like think about these interactions?
Okay, I'll start with the second one. Both of these are very dear to my heart. So the second one is suggesting that, first off, adaptation and this short-term plasticity effect are very different. So we know that. We know that adaptation you could think of the short-term monocular deprivation as a form of de-adaptation because you're presenting one stimulus, one eye, with lower contrast stimuli than the other. However, adaptation would be, number one, orientation selective. And here we are seeing an effect that is not orientation selective. Number two, adaptation typically transfers across eyes, at least a good portion of that. And by definition, here we're seeing something that goes in opposite direction between eyes. So at least these two are very strong indications that what we are looking at is not simply a de-adaptation effect. Now, time course of the effect, again, quite different from the adaptation. The short-term plasticity doesn't seem to vanish or accumulate over repeated sessions. Note that the effect on amblyopia is achieved by repeated adaptation, but it's not an ocular dominance shift, it is an acuity change. So there might be other mechanisms that are set into motion by these short-term effects, even though they are short-lived, they have long-term consequences. Adaptation, on the other hand, tends to vanish as you repeat it, as you suggested. Having said this, nobody has ever tried to do the short-term manipulation for multiple days. So the longest we've come is 10 hours. And what we see in 10 hours is essentially the same as you see in 2 hours. Nothing changes much. And it will be interesting to see if we could extend this at least into the days. But so far, we don't have the data for that. Now, back to the delay manipulation. I'm afraid I forgot the question. Would you mind repeating it, the first one?
Have you tried doing something that is not consistent across the full visual field?
Thank you. Yes, this makes a very important point. The delay manipulation is not consistent across the visual field in the sense that you only ever get a conflict between the eyes for the moving parts in the visual field. So anything that is static is perfectly compatible between the two eyes, and it's like you never did anything to that. You only have, we computed that roughly, around about 10% of pixels at every time point where there is a conflict between eyes introduced by the delay. And that's because it is a delay. With a prism, for example, you would have a complete mismatch across the entire visual field. Here, with only a 10% mismatch, we have the same effect that we would have by occluding the eye completely. So this tells us something about the non-graduality of this effect, I would say.
Got it.
Thank you.
Lovely. I see that. So I've got another question. At the moment, nobody's raised their hand. So I'm thinking about kind of like closed loop. Now I know that some things you've shown are also happening when people are just patched and they don't have the activity or the the action related part, which is eye to hand coordination. But still, it could be thought about that the pulvinar may be playing a role both in the inputs and in creating through the superior colliculus pulvinar and going, let's say, to MT or to the frontal eye field, and also in the updating of the information streaming through the, let's say, the cortex. So it's related to saccades and motor planning, oculomotor planning, and also to the inputs. And I'm wondering if because of that, like chopping the loop would maybe there's like some chase to keep up like a diet. You are deprived of something, so you want to get as much as you can before things and maybe LGN is more input-ish. I don't know, maybe that's all. But I'm thinking if that has some, maybe.
Yeah. No, I like this a lot, and this is actually the project that we are pursuing right now.
Okay.
So the active vision. Active vision is necessary to see the effect of monocular deprivation. It is not necessary to see the effect of monocular patching, although there is some evidence that it can enhance that effect. So the way I would piece this together is by assuming that whenever you are acting, you are producing the visual changes. So not only there is an expectation based on general context, but there is also a corollary discharge that gives you the expectation. And I'm doing this with my hands, which obviously have lots of degrees of freedom, but if you did with your eyes, then you would have a much stronger expectation based on a more established corollary discharge circuit. So we could take this whole set of manipulations and connect that with the eye movements manipulations that we're very interested in. And that's, like I said, this is exactly where we are headed right now. Thank you.
Wonderful, wonderful. So if there are no more questions, we will give you again a very big applause for a really wonderful talk. And thank you so much for joining us and for sharing these exciting findings with us. And next week we will, oh, Jeremy, this is lovely. How do you do that?
By mistake, actually, but apparently, if you are in the reactions,
Aha,
oh,
a
I
new,
see. There's a stream that are send with effect, so you can
Oh, wow.
aha.
do these
Okay, I'm going to
things,
try
and
that out. Amazing. Okay, great.
we can have
So,
just endless fun
so yeah,
here.
So next
Thank you,
week,
Paola.
Thank you.
yeah, thank you so much. And next week we will have here Melissa Vo.
Oh,
And
good.
but it's going to be 1 hour earlier than usual. So I really hope everybody can join, and especially early, right, like from the US, which is already early for them. But you know, make an effort if you wish. So yes, we will have Melissa here. And thanks so much for joining us again, Paola. It was really great
Thank
having
you
you.
very much for the feedback and the questions. Thank you.
Thank you so much. Bye-bye.
Bye.
Bye, everyone.
Bye.